There's something in our brain that allows us to recognize someone's age. Have you ever noticed how interesting that is? That we have like this innate ability. Like if you were to look at someone, you're generally going to be able to guess their age within 10 or 15 years. It's something that allows us, maybe from a biological perspective, to understand how old someone is. Maybe they're viable as a mate, maybe they're not, based upon their age. Maybe it's truly biological like that. But it does go further than that. You see, we also have to understand what causes aging, because I think there's a common misconception in the research, or at least how the research is understood with aging. We think that it's just getting older and dying, but there's a lot of different pieces that go into aging. And that's why you can't just look at someone and say, okay, they're 76 years old, so they're old. That's their chronological age. If that 76-year-old person has the genetic makeup and has taken care of their life and has the right lifestyle to actually be younger from a biological standpoint, who's to say that they can't be 76 years on a calendar, but maybe are 55 years old as far as their cells are concerned, right? So we're going to break down what's called biological age, and it's pretty complicated, but I'll give you a really distilled version so you can understand, and also some very practical things that you can even try, and even an interesting website that is produced to input some of your like blood chemistry data to actually figure out your biological age. So let's go ahead and break it down. So when we look at the scientific term aging clocks, it's looking at multiple different things. Now, to make a very, very complicated thing very simple, I want you to imagine for a minute that there was only one thing that causes aging. Now, it's funny because back in the 90s, we actually thought that this one thing was the main thing that caused aging: oxidative stress. So let's take oxidative stress as an example. If we were to know that oxidative stress were hypothetically the only cause of cellular aging, it would be pretty easy for us to look at biomarkers and find, oh, the biomarkers for oxidative stress are high. This indicates that this person's biological age is probably high, right? It would allow us to determine their age much easier because there's only one data point. However, it's not that simple. There are a lot of moving pieces, and we're going to break it down more. Now, what's interesting is there was a man named August Weismann who a while back proposed that maybe aging was genetic, like puberty. Maybe like we're pre-programmed to go through puberty at a specific age. Maybe we're pre-programmed to die and age at a certain age, which does make a lot of viable sense. However, when you start thinking about what's called like evolutionary pressure, there would be a lot of evolutionary pressure for gene mutation and all kinds of things. So there's some flaws to that theory, although it still does hold a little bit of merit. Well, let's look at the alternatives when it comes down to biological clocks, because there's no singular process. We're not living in the 90s. This is 2022 as of this video, right? We have a lot of different processes that are associated with aging. Now, the journal Cell had published a study a while back that indicated that there are 9 hallmarks of aging. They identified 9 things that are associated with aging. In this case, they looked at cellular senescence. They looked at stem cell exhaustion, so how long it takes before stem cells just aren't really being produced anymore. It's a very important piece, so that's one of them. And then also altered cell communication was a very important one. And this includes inflammation, because inflammation alters cell communication. They also looked at mitochondrial dysfunction. So basically our energy powerhouses being unable to convert the fuel into energy efficiently. Okay, this highlights the importance of like mitochondrial biogenesis. They also looked at genome instability, like methylation of our genes and how our genes just become unstable. Deregulated nutrient sensing. So this means like becoming insulin resistant, not being able to sense nutrients is a hallmark of aging. A loss of proteostasis, which is where the proteins are no longer folding and unfolding properly. This highlights the need for autophagy occasionally to help chaperoning proteins allow this to occur. Also telomere attrition, right? When the telomeres start to shorten, that's a big hallmark of aging. And lastly, epigenetic attrition, where our epigenetic system just isn't expressing genes properly. So some things are downregulated, some things are upregulating. Anyhow, there's a lot of moving pieces. The point of me mentioning the hallmarks of aging are what we're looking at now. So 9 big variables that give us our biological age. The problem is that the current aging clocks don't really give us a universal number. They're not looking at all of this, they're looking at a subset. For example, we'll look at like epigenetic age. So the effects on the epigenetic system are going to give us a number there. So let's break down the most common types of biological aging clocks that researchers look at. The first one is going to be the epigenetic clock. And this is one that is looked at probably the most. It's probably the gold standard as of right now. So there was a 2013 study published in Genome Biology, and this really highlighted the epigenetic clock. And what it looked at was what is called DNA methylation. I've talked about this in other videos, but essentially it's where methyl groups attach to the DNA, and when they attach to the DNA, they silence a gene. So basically they can silence a gene from doing its job. This methylation, DNA methylation, can be a very big problem. That's really what the epigenetic clock is looking at. Essentially, what it allows researchers to do is estimate the age of stem cells and also look at stem cell exhaustion. Now, there's a lot of nuancy stuff that's beyond my pay grade, but if you look at some other data, it's pretty interesting. There was a study that was published in the journal Clinical Epigenetics that looked at epigenetic age, and it looked at something called age acceleration. So age acceleration would be if you were 50 years old and your epigenetic clock said you were 55 years old, that would mean that you had an age acceleration of 5 years. It means your biological age exceeds your chronological age. Now, if you went back the next year when you were 51 years old and your biological age was 60, that would mean that you have a lot of acceleration, a lot of velocity with your epigenetic age. So that means that things are not so good. And what they found in this particular study is that the age acceleration was linked to mortality. More age acceleration did equate to more mortality. And then you look at things, for instance, in this particular study, they found that smoking does in fact increase epigenetic age. Now we do know from other studies that smoking can increase mortality, but now we know indirectly that smoking increases epigenetic age, and epigenetic age, when it's accelerated above your chronological age, can influence and increase mortality. Now there's a study that's published in Genome Medicine that found that this form of biological age, it's very accurate, but it's also very context dependent. Okay, so we don't always know, like if you have an increase or you have methylation of different groups, sometimes it can be a good thing, sometimes it can be a bad thing. So generally speaking, we get an accurate picture, but it is somewhat context dependent. So that's why we're always trying to refine this. After today's video, I want you to check out Qualia Focus. This is a cool product because it's not like a crazy over-the-top nootropic. It's a focusing agent, something that helps you kind of get in the zone. It's something that I like to take when I film, and I literally feel like I get in the zone when I take it. So there's no proprietary blends, no weird stuff, and they're the only brand that is using complexity sciences to formulate. So they're really putting their money where their mouth is when it comes down to proper scientific formulation. So the cool thing is, if you're really interested in sort of leveling up how your brain feels and just getting that little like perk up and focus that you need without the jitters, without feeling over the top, you've got to try Qualia Focus. So if you go to the link that's down below, you can check out how you can save 50% off your first purchase, but also another 15% by using code Thomas because you're watching this video. So we're talking a potential 65% off your first order of Qualia Focus. So again, it's something that you just want to use if you're trying to just get in the zone. You could use it daily, you can use it sporadically when you feel like you need that extra kick in the pants. But the cool thing is there is a 100-day, 100% money-back guarantee. So if you're not digging it, if you're not feeling the way that I say you're gonna feel, you're not feeling what I feel, then by all means, just return it for 100% of your money back. So that link is down below. You wanna use code Thomas to save that extra 15% on top of the potential 50% that you can save by hitting that link down below. So that's in the description down below underneath this video. So check them out after this video. So that's how we kind of evolve into the next one, which is sort of a transcriptomic clock, right? Where you're looking at the transcriptome age, so to speak. So let's break that one down. So this was highlighted in a study in Nature Communications. Okay, so when you have a gene, this gene gets transcribed into what is called mRNA, messenger RNA. Once you have the mRNA, ribosomes transcribe the mRNA into specific proteins. So what we're looking at with the transcriptomic clock is we're trying to identify what messenger RNA is really associated with age. Now, a transcriptome is a term for all the mRNA inside of a cell. Okay, so unfortunately, there's a lot of room for error there because mRNA, that's a big field, right? There can be all kinds of different things. We'll talk about some other clocks that poke some holes in this in just a second. The good news is there are multiple causes of aging that can be looked at. So when you look at the data, they found there were 1,497 genes that were associated with aging when they looked at the transcriptomic clock, okay? Now they found that 897 of these genes were downregulated and 600 of these genes were upregulated. So this means that certain downregulation of some genes affects aging and certain upregulation of some genes affects aging. Let me explain what that means. So they found that the upregulation of pro-inflammatory genes was associated with age, which confirms how chronic inflammation seems to increase with age. That's called inflammaging, right? They also confirmed that downregulation of mitochondrial biogenesis, downregulation of just mitochondrial health in general, genes associated with that, were also associated with aging. So that confirms the need for like fasting and things like that to improve mitochondrial biogenesis. So you see how some things get upregulated, some things get downregulated in association with age. Now the interesting thing is that the precision isn't exceptionally high. It's like a mean error of 7.8 years. So it's not super accurate, but the relevancy is high. What I mean by that is it gives us a lot of detail into specific things. Again, 1,497 very specific genes that can be looked at that are affected by this clock, right? So when we look at that, we get a lot more detail and relevance. We just don't have 100% accuracy or closer to 100% accuracy like we might get with an epigenetic clock or some of these others. Then we have what's called the proteomic age. Now the proteomic age takes a look at the mRNA and says, well, wait a minute, there's a problem. Because the mRNA, by its very nature in a human body, can be spliced. So there's something called a spliceosome. So the messenger RNA that we already have can be spliced within the spliceosome and turned into different sequences, okay? So it can be just have all kinds of different gene sequencing occurring within our body. So when we look at mRNA, we don't get the full picture. What proteomic clocks look at is basically almost algorithmically sequencing genes. So very complicated if you ask me. It's not my area of expertise, but essentially it's a way to get a more accurate picture. So basically you're bypassing the whole errors with the mRNA by being able to sequence the proteome a little bit more, giving you a more accurate picture of where the mRNA might have certain upregulation and downregulation. Then there's one that's really interesting. This is the blood biochemical age. It's not the sexiest out of all of them, but it's actually the most attainable. Okay, what this one does, it takes a look at 19 blood biochemical markers. So things like HDL, like glucose, just various blood markers. And algorithmically looks at those and says, okay, well, based upon blood biochemical markers, this is what your biological age is. You can go to aging.ai and literally input your own blood work in. And that's not a sponsor or anything, it's just a free system, aging.ai. So if you entered your blood work into there, it would give you a pretty detailed biological age out of that, which I think is just really cool because it's super attainable for people. The nice thing is the mean error is about 5.5 years. So pretty accurate data just looking at 19 simple blood markers. The next one is one that's really interesting because it's called photoaging, which is just like the name implies. It's actually the most accurate, which is interesting. Accurate all the way down to 2.3 years. And there was a study that was published in the journal Aging that demonstrated that when you look at the corners of the eyes in extreme detail, you can algorithmically figure out someone's biological age, which is interesting because we have a lot of these detailed things that we're looking at with cellular senescence, with mRNA, with DNA methylation, but sometimes when it comes down to just looking at the corners of the eyes, I know you're all looking at the corners of my eyes right now, trying to figure out if I'm old or not. Point is that it's more than what you could just see with the naked eye, right? There's specific things that are looked at. Unfortunately, the algorithm isn't available to the public, so we can't just upload a picture of ourselves to see our biological age, but it does go to show that the way someone looks does sometimes tell us a lot about their overall biological age. It also tells us that someone really can have some reversal with this process if they take the right lifestyle approaches seriously. So at the end of the day, forget how old you are. You should never celebrate your birthday, ever. I'm just kidding. But you really shouldn't think about it as a number like that. You should think about how you feel and your biological age. So as always, keep it locked in here on my channel. I'll see you tomorrow.